get the refreshments, and I am pleased now to introduce to you Dr. Michael Nair from Shimadzu, who are our platinum sponsors this year. So Michael, yours, microphone.
Thank you. Good morning. It's nice to stand in front of you all today for the latest imaging meeting. I've been coming here for quite a few years, and it's been great seeing Malcolm talk about all his students and getting everyone to come up and present. So this is my first time presenting here. I've been in MALDI for about 11 years now. I've spent 10 years working with MALDI manufacturers and helping develop applications, support customers. And I'm really pleased to be able to present some information on affordable MALDI imaging, especially as Malcolm was saying, one of the biggest challenges we all face is trying to get funding. This should hopefully help get you guys more funding by doing the preliminary work on a more affordable system. So mass spectrometry imaging is very expensive. There's no getting away from it. We've got DESI and LISA, which is expensive, MALDI imaging, which is very expensive, and SIMS, which is really, really very expensive. And then there's cost of looking after the instruments. So what do we need to do imaging? We need an imaging-capable mass spectrometer. We're trying to let you know that there's another way that we can do this on a more affordable benchtop MALDI instrument. We've got the MALDI-8020, which is a linear-only MALDI-TOF, and we've got our MALDI-8030, which does both positive and negative polarity. But with things like lipid imaging, the MALDI-8030 is a preference because otherwise you really won't see your lipids very well at all. You'll need a microtome, you'll need imaging slides, your matrix application, and data analysis software. And with that, you should be able to get on with some pretty simple imaging experiments. So when we visited ASMS conferences, we saw that a lot of people have very high-end MALDI-TOF mass spectrometers that have the latest Ultraflex or Rapidflex, and they'll present data where they're looking at intact masses, and they'll be looking at intact masses at about 50 micron spatial resolution. We thought, well, our benchtop can do this. This little benchtop's got fast laser in it. It's a fraction of the cost of one of these systems. So we went back to Shimadzu and we thought, let's see whether we can get this to work on our system. We started development to create a package that could give you high quality imaging, but on an entry-level benchtop. Oh, can we go back one? So this is a screenshot from the first experiment we did back in 2017, where we're just evaluating, can the system actually do what we're asking it to do? So we're looking at the laser, the reproducibility, and this was enough to get us quite excited. We went in from this thinking, okay, well, we can do the basics. We can obviously ablate spots in a reproducible pattern. We then went on and started doing some development work to get things into a reproducible and consistent manner, because without reproducibility, there's just no point. From there, we took some time, but by 2023, we had a system that was capable of delivering images like this. And we were very happy when we started getting data like this off our system. So what do you need? Why is the benchtop useful for your lab? So I don't know whether you've seen many imaging systems, but they're generally quite large. The timsTOF, you need to have quite often your roof changed so you can fit one in. The Waters MRT systems take a bit of space. This little benchtop is quite small, so you can fit it in smaller labs. And with space at a premium, this is actually a really useful feature. It's got a fast laser. Now, we're not talking about 20 kilohertz, but we're talking about 200 hertz, and that's fast enough for a lot of entry-level kind of work. And you can obviously do dual polarity. It's not very noisy. A lot of mass spec labs, you won't notice anything because you've already got all the pumps, you've already got all the fans. But if you're starting out and you've got a smaller lab, having an instrument that doesn't make a lot of noise is always a nice thing. You've got the ion optics, which are very wide. They allow great ion transmission, so you're getting better sensitivity than you would do with more restrictive ion optics. The AON detector, I think Malcolm was talking earlier on about you're talking about lasers not lasting more than 2 or 3 months. The detectors are another thing that take an absolute beating when you're running constant streams of ions up the flight tube. So that, combined with being able to clean ion optics after running some messy samples, you're talking about a system that it's facilitating you doing this kind of work with minimum downtime, minimum engineer time. And now we've released Easy Care, so you can actually take the ion optics out, you can give them a good clean and stick them back in, so you're able to restore the performance back to what it should be. So with this, you don't have to be afraid of researchers using an instrument. You don't have to worry about the diva instrument that somebody's going to mess around with and then you're going to be stuck for 3 months waiting for an engineer. You're actually going to be able to do this yourself. What is the worst they can do? But that's not a challenge, because researchers can do some very interesting things. So, for example, don't use loose powders, wet samples, or carbon black. These things are known to kill MALDI-TOF mass specs. So what do you need to get started? Well, if you've got the benchtop MALDI already, then you just need an imaging kit. So this comes with the slides and adapters and so on that you need. You've got this here, which allows you to scan your slides on a scanner. You've got reusable slides, you've got slide masks for when you're applying your matrix, and then you've got these glass slides and an adapter so you can load it in the instrument. With that, you also get the activation to the imaging wizard and ion view, so you can actually set up your experiments and analyze your data. So what else do you need? You need to apply matrix. We've got two options. We've got a sprayer, because this allows you to do things like spray your matrix, obviously, but it also allows you to add a derivatization reagent, or it allows you to digest your proteins. Then we also have the ion layer, which is the class-leading sublimator, giving you the finest crystal sizes for where spatial resolution really is critical. You don't always need this for a benchtop workflow. You might say it's overkill, but the joy of it is it makes applying the matrix very, very easy. You just weigh out your sample in a boat, stick it in and press a button. It's really that simple. So there are two different instruments for two different jobs, really. So with ion layer aero, you've got humidity control. I've spoken to a few imaging specialists who find problems with sprayers generally is that getting the reproducibility from the spray can take quite a bit of effort. By displacing the internal atmosphere with dry nitrogen, no longer a problem. It's a stable condition, so you're getting the same kind of spray every time. And another issue that happens with sprayers is clogging. This system has a built-in system to clean the nozzle, so you're getting perfect spray every time. So this is compatible or validated with DHP, 9AA and CHCA. You're also able to use Gerard's reagent and you're able to use trypsin with it, so it should make your life a wee bit easier. Then we've got the IM layer. Sorry for the terrible joke at the top. That's all my doing. So you just load the sample, load the matrix, load the method and walk away. It's so simple, I can use it. It is very reproducible. I've done a lot of work where I just prepare my sample, bung it in with the matrix and you're fine. How am I looking for time? Good. The sublimation time, temperature and layer thickness can all be user controlled. And actually one of the really nice things is you don't need to use MALDI grade matrix. You can use the really, really cheap stuff. And the reason for this is a few different things. You've got a shutter that blocks off the sample, the matrix boat. So until the temperature is right, it's stopping any early sublimation material coming up and contaminating your sample. It then moves aside, allowing the right temperature of matrix to be sublimed, coating your sample for the defined period, then shutting off. So you've got really good control of how long you're actually coating your sample. So the sublimation generally will purify your matrix, but that shutter means that you're only getting the matrix that sublimes at that particular temperature. If that's not enough control for you, there's also a laser that can measure the thickness of the matrix on the sample slide. If that's of concern, you can use that. I've never found I needed to yet, though. The IM layer has got methods preset for CHCA, DHBA, 9AA. You can develop custom methods where you can change the temperature and the duration if that's what you want to do. But yeah, it's the first commercial MALDI sublimator as far as I know. I'll let Malcolm correct me if I'm wrong. You've got the smallest crystal size. We've recorded crystal size of 50 nanometers for DHB, and it's got the highest reproducibility because you've got all these features allowing you to have that really tight control over sample sublimation. So you can see this is where you attach your matrix boat here. Your sample fits on a carrier up here. Then you've got software. So to get started with your imaging experiment, you need to co-register your sample with your MALDI target. So we've got the software for this in MALDI Solutions. You can co-register your glass slide, select the area you wish to analyze here, and then you'll be much more efficient with your time. And then you need to analyze your data. When you buy the imaging kit, you get IonView as part of the package, and it's not the fanciest piece of imaging software. But if you bought imaging and you didn't have something you could analyze the data with, you'd be a bit sore. So this will do the job for you very well. You can, as Malcolm was showing earlier on with going through the various masses and seeing the various distributions, you can do that, and it's one of the features I particularly enjoy using when I'm playing with it. And it's included with the imaging kit. Then if you're feeling a bit more flush and you need to do more statistic work, you've got image reveal, which allows you to do a lot of the advanced imaging features you would hope to do on an imaging system. So image reveal allows you to do basic statistic analysis, principal component analysis, partial least squares, image classification, similar image extraction, segmentation analysis, arithmetic options, quantification, and multimodal imaging. So it really is probably one of the more versatile pieces of software that's kicking around for this kind of work. So where would I be talking in front of Simona's meeting if I don't show a fingerprint? So some of the first work we did when we were getting the system quite close to validation was looking at fingerprints, and these images show that we were very happy with the system. We were able to see really nice resolved images, and I believe these are probably my fingerprints. And we've highlighted a few masses there, not of any particular relevance, but they do create a rather nice fetching image. So we've got finger ridge interspaces at MZ549, finger ridge features at 829, and the sweat glands at 375 and 567. Likewise, if you're developing an imaging system, where would it be if you don't look at some animal brains? So we've got some images of the lipid distributions in rat brains. We've highlighted a few phospholipids that you can see in these images here. So here is a digested protein that we've got. So we're looking at a much larger protein that you wouldn't otherwise be able to look at by digesting it with the IM Aero. Then you're able to start looking at these kind of proteins on your tissue sample. But it's not just the team in Kratos who've been working on this. Our colleagues in Japan did some rather interesting work looking at PET films and looking at how they vary with exposure to different heat conditions. So with the benchtop system, we were able to look at the presence of the cyclic trimer of PET under various heat conditions. So the cyclic trimer there has the mass of 599.3 if you've got the sodium adduct on it. And here we can see the intensity of the peak for this trimer increasing with exposure to no exposure, 4 hours at 175 degrees and 18 hours at 175 degrees. So you can see how the polymer itself is releasing more of the cyclic trimer with exposure to heat. And we've been working with Nottingham University, been working with Sidra Raman to develop a system to, well, looking at exogenous metabolites from bacteria. She and her group have already been working with Lisa and Sims and Desi and Reams to look at the metabolites that are observed when bacteria are present in competition with each other. And we've taken this work a little bit further and consolidated the identities that we've got from Sims with what we can do with bacterial imaging. But as you can see from this, it wasn't immediately successful. The first problem we had is how do you have the agar on the slide and not have it falling off? But with a bit of emery cloth and a bit of practice, we managed to get the agar to stay on and started being able to do work where we could culture cells on the MALDI targets themselves and then analyze them after that. So we started off, we ended up getting one of the engineers at the factory to design a slide mask, had the FlexiMask slides. Obviously this one's not been abraded yet. And then we pour the matrix onto the slide mask. We use polycarbonate discs to put on the agar so that the bacteria can access the nutrients and the moisture from the agar and then we grow the bacteria on those. When we're finished with that, we remove the polycarbonate discs, just leaving the metabolites that are in the agar. We place a slide into the IM layer, run it for about 20 minutes by pressing the button and walking away. And then we've got a sample like this. It's ready to go in the mass spec. So we've got images that Sidra has acquired where we've got the standard Pseudomonas aeruginosa sample here, and we're looking at these different metabolites. Some of them are not the most intense, but we're seeing these metabolites present in these samples. And what you're seeing with the Staphylococcus aureus is you're seeing a reduced expression in some of these metabolites, and then when you've got the Candida albicans, you're not really seeing much there at all. We've also worked with Southern Illinois University. I think it was back in 2022. I was across ASMS. Forgive me if that's a COVID year. And I met Kevin Tucker, and we had to talk about benchtop imaging without talking about benchtop imaging because this was very confidential. This is something we were developing under wraps, and we couldn't tell anyone about it. But we had a very good conversation, not about benchtop imaging, while really trying to suss out whether he would be a great collaborator for us. But he was really interested in this because he wanted to see an affordable imaging system. So we were both talking about the same kind of thing. So he's done some work. He's found out the kind of exposure levels of statins you might expect to see in soil. So he's doing some environmental work. He's done some quantification by LCM-SMS. And he wants to see how these distributions change actually within the mouse. So this is a great example where he's using the MALDI in collaboration with a pre-existing workflow for quantifying. And this is rather an elaborate process, but basically he's freezing the mouse, he's extracting the proteins, and then he's looking at these on the LCMS. And then he developed a creative method for getting the worms to be a size and shape and reproducibility for MALDI imaging. So he was very proud of how do you get a worm to fit a MALDI target? Well, you stick it in a beer bottle cap. So he freezes it in nitrogen, microtomes it, does an ammonium wash, and then we're able to get images. Some of them are much better than this one. But we're seeing lovely isotopic resolution for some of these smaller masses. So these are the internal standards he's used. So we've got, I'm sorry for my pronunciation, atomvastatin, lovastatin, and simvastatin. And here what we're seeing is that there's localization of the lipids and related metabolites for the different statin exposures. So they are accumulating in these worms, and we're able to see how they're distributed within the worm's anatomy. Some work that's being presented by Caroline Jones is on using MALDI high-plex immunohistochemistry tags to label large proteins. So we're able to multiplex our image samples using multiple antibodies and tag larger proteins that you wouldn't normally see in MALDI imaging with these mass tags. So the sampling is quite simple. You might use tissue sections, you expose them to the antigen, you cleave your antigen, you apply matrix, and then you get your images. Very straightforward. So masses like 23,000, 75,000, I think you would really struggle to see intact masses like this on most mass specs. But by using these mass tags, we're bringing them into a range where you're getting isotopic distribution, and you're able to clearly identify these peaks in the data later on. So, I mean, these are some astoundingly large proteins that we're looking at here. So here's an example mass spectrum. You can see the mass tags are showing up very clearly here. They're not struggling for signal. And if we look closer at this, we can again see the isotopic distribution for some of these proteins that we're looking at. So these are not the proteins we're looking at, just the mass tags. And when we look at this in IonView, we can clearly bring out these images showing the different distributions of these proteins within the tonsil sections here. So this is all positive ion mode, it's 50 micron spatial resolution. And you can overlay these images as you can see here. If you use IonView, you can multiplex 6 proteins or more in IonView, so you can get much more image-rich and more pleasing images for your sample. You can look at more different protein expressions over your sample. And with the imaging, we can see the epithelium, we can see the extracellular matrix, we can see the lymphoid tissue and macrophages. This is a lot of great information you can use for identifying diseases, and with the statistic tools of image reveal, you can then compare this to healthy sections, disease sections, treated sections, and identify what's being differentially expressed. So with that, I would like to thank you all for listening, and I would like to thank my friends at Nottingham University, at Kratos, and Southern Illinois University for letting me use their data and present it here for you today. And I hope you've enjoyed the talk.
Thank you very much, Michael. Got a couple of questions, but you first.
Hi there.
Over there.
Did you account for any height differences when you were imaging the cells of the world? Was it quite flat?
You can use lock mass so that you can then adjust the mass spectrum for any differences in topology. Hello there.
Is that how you analyse the insulator as well?
Sorry?
The lock mass on the membrane. Didn't you expected the mass to drift a bit when you're analysing the membrane?
The membranes, yeah, I mean, we expect masses to drift when you're doing imaging because no sample is really fully flat. Everything dries differently, so yeah.
Yeah. So lock mass, yeah.
Oh no, hang on.
Sorry, recrystallization, is that possible to automate that on the IM layer?
Recrystallization is still a manual process, but we can give you tips and advice on how to do that, but it usually involves a box with some water in it.
HTXL sublimation as well.
Thank you.
I just like that.
Hello there.
For bacterial imaging, what was the resolution?
For the bacterial imaging, the resolution wasn't key, but we were doing about 50 micron. So the work had also been done on DESI, but I think we were getting, with their DESI setup, we were getting much better sensitivity. But as you can see, there still wasn't a huge amount of things to look for there.
I'm going to ask you more of a philosophical question. We were just discussing about this in the break.
Yeah.
Obviously, these benchtop systems have different specifications, different capabilities. And so it might be more appropriate and actually better if your finances are particularly big. Where does Shimadzu see the placement of these benchtop systems where we don't need such a high spatial resolution or maybe such high mass accuracy? What is your sort of vision for placing these instruments? What kind of environments, what kind of end users, what kind of applications?
We're looking to find as many new areas to explore as possible, but certainly I think we've seen some work where people have used the benchtop for looking at seed coatings, so agricultural universities, food labs, when you talk about food contact and exposure to films, are able to see things like PFAS, for example, migrating into foods from the films that they're packaged in. What other kind of interesting things can you find? Can you find degradation in fats? There's a lot of questions, and if you want to speak to us about this, then I'm happy to answer your questions and see whether it can be done. And if I can't answer it, I can also speak to my friends at Kratos, like Tom and Matt, and got colleagues in Germany and people in Japan. So people are very much interested in expanding the areas where we can do multi-imaging. So this is why forensics is interesting. This is why the work with the earthworms is interesting, and the work with microbial metabolism. So if you've got something you're interested in looking at, come and speak to us. We'll see whether we can help.
One more question there. Why we should think that matrix sublimation is better than spraying?
It's not always better, but it's different, and it will give you the finest crystals. But as our friend over there at Verulam mentioned, recrystallization will help you get more sensitivity out of a sublime sample. But sometimes a spray will be a better option because it's a more wet more wet means of applying matrix. It will pull out more of your analytes of interest. Sometimes the spatial resolution, you really want to go for sublimation, and sublimation is also dead easy once you have one. So horses for courses. You choose a method that suits you, and you'll find different techniques will give you different results, but only in terms of sensitivity and spatial resolution.
It doesn't apply to all master charges, to all range of molecular sizes.
think you had a very good range of questions.
Yes, I think it's
Thank you
excellent.
very much for
Thank you
your
very
talk.
much.
Thank you.